All right. I'm Howard Halpern, and I've worked in EPR imaging at these very low frequencies for a long time. I've had the opportunity to know Josh Biller for a dozen years from his graduate work with Gareth Eaton to his experience not too far away in Boulder, Colorado at NIST, and now with TDA Research in Golden, Colorado. He is a very, very insightful, not quite so young man now. He's obviously got a lifetime or an academic lifetime commitment to EPR and to some of the issues that we're all dealing with. And I'd like to just, with that general introduction, introduce Josh Biller. Take it away, Josh.
Okay.
And I will stop share.
No. All right, can you all see my PowerPoint presentation in presentation mode? All right, so I want to say thank you to O2M and to Merignani for organizing this webinar series. It's excellent to hear from all the speakers who've come previously, and you're going to note in the presentation, I point out some of the ones that are coming in the future that you should definitely take note of. And I have to say it's been especially nice in this past year of pandemic when conferences and such have been canceled to still see updates on research and to get the community together thinking about what are hard problems as we try to transition from preclinical to clinical EPR imaging. But as I end with this presentation, they don't give the easy problems to the smart kids. So hard problems is what we're gonna work on. Okay, Mary and I did advance to the second slide. So I have two slides for what I mean by the preclinic. The first one just represents a way that I think about EPR imaging and EPR in general. I was brought up through the Eaton Laboratory where we developed methods, rapid scan, pulse, CW methods to augment CW or EPR imaging on animals that was being undertaken in the Chicago lab under Howard's direction. But it turns out that there are other applications that care about things like oxygen concentration and pH and redox status. Corrosion is an example in the in situ material science field. And the main take home from thinking about this is that there are parallel paths for developing useful tools for preclinical and clinical imaging that could maybe go through materials science routes without the extra rigor of maybe clinical trials or FDA or having to work on critters. That's an entirely different talk. And the one we're going to talk about today is this, preclinical to clinical. But being a chemist, I do tend to think of biology as just a more complicated material science problem. So here's the outside the preclinic I'm going to be talking to today. And we have two mice. One's asking the other if they got him all fixed up. And he says, yes, cancer and Alzheimer's today, back tomorrow for a cure for my diabetes. So we've cured many diseases in mice. It is unfortunate we cannot think like men and women and solve only the problems of mice. We have to translate this out of preclinical work and into the clinic. So I was a big fan of Hal Schwartz when he gave his presentation and basically said in the first couple slides, here's the big idea in case you fall asleep through the presentation. The presentation is set up to explain why I think this image sequence is exciting. And the take-home points are we can construct a nanoparticle, we can attach a paramagnetic imaging agent, and we can encapsulate it with a biocompatible polymer. I did not send this proposal off to the NCI with RADI-sense as the name. It picked it up mysteriously through the NCI reviewing process, and I just kept it. The oxygen sensitivity of the agent is maintained, and you can use it to capture quantitative PO2 maps in mice, just like you can with the free agent. So if you kind of zone in and out or come back, this is the main take-home point. So the overview, 2 slides answering the question, who is this person talking to me? And as Howard alluded to, I am a not so young man anymore. I have a 7- and 4-year-old who keep me moving quickly, but also remind me that age does march forward. What is the company he's working for? I did want to highlight the project team, both at TDA and amongst our collaborators for this. I have been, it's been very interesting for me. This is project I'm describing as the first one where I'm formally the primary investigator. And so I've been dealing with the reviewer comments and engaging with the program director, and the questions I've gotten about moving EPR imaging into the clinic. Now, the people who've been doing this for much longer than I have are maybe going to chuckle or nod their heads or say, we answered this a long time ago and it's still coming up. But all of these interactions I'm describing have happened in the last year. So these questions that people have are still relevant and things we have to overcome as a community, but things that I think we can. I am going to talk a little bit about some of the results of our prototype clinical EPR imaging agent. It was an imaging agent designed to be ready to go into the clinic, and designed to be something that you could put into a clinical trial. And then summary thoughts and conclusion. So who is this Josh Biller? And why did he just give us in pictorial form his CV for the past 15 or 16 years? When I was preparing this talk, and I was also reflecting on comments that Howard had made about the need to do quantitative measurement, that resonates with me. And it resonates with me because no matter what I've been doing for the past 16 years, that's been a major part of it. I started life at the Mayo Clinic as a clinical chemist. We used ICP mass spec instrumentation to do trace metal analysis. And so I was introduced to things like a normal range, standard deviation, quality control, and the need for very high reproducibility. And I was also introduced to the fact that you can make measurements that others deem impossible. We once had a development tech at Mayo present at a conference, and a professor from Texas who was in the ICP-MS world stood up and told our development tech that what we were doing was impossible, as the tech was presenting data that we were doing it. And you do that by knowing what the measurement is that your instrument is making, and making it in a quantitative and reproducible way. So then I went on to DU, and people like to throw the phrase around, well, so-and-so wrote the book on some topic, but my advisors did. And Gareth and Sandy Eaton put it together in a very just the facts, ma'am title, quantitative EPR. And so that quantitative measurement was reinforced. I postdoced at the magnetic imaging group at NIST. That's where I got into low field double resonance techniques and solution over Hauser DNP, which really exposed me to low field nuclear magnetic resonance, low field imaging. There's some things that we can take from that as the EPR imaging community. And then also the quantitative MRI effort there, the creation of phantoms, standards, driving at normal ranges for T1 and T2 in tissue, which is not something that exists currently. And then for the past 3 years, I've been at TDA. And I've done a whole number of things, one of which I'll tell you about today. So again, we need to do measurements quantitatively so that we can repeat and believe the measurements. This is pulled right out of Howard's presentation. We can diminish the effect of confounding variables. We do calibrations and baseline corrections. And we can use reference materials or objects to anchor our measurements so we can clearly answer the question in this instrument response, what if that corresponds to reality? And then one slide here about TDA research. We're not widely known. We don't advertise. We stay busy. And when I always ask about, you know, kind of advertising campaigns, the the comment sometimes is, well, the people who need to know us do. It was founded in 1987 by three former scientists, 2 engineers and a scientist from what has become NREL in Golden. There's 104 employees currently. 90 of us are technical staff, 30 of us have our PhDs either in chemistry or chemical engineering. The remaining 60% have either bachelor's or master's degree in chemistry, chemical engineering, physics. And we are a professional R&D firm. We just like solving difficult problems. We have a number of chemistries and things that we've developed and then licensed out, ranging from this over here in the bottom right-hand side is a picture of a large facility. And so there was a process developed at TDA to make large-scale fullerenes, and this facility could output 30 tons per year, all the way down to specialty conducting polymer chemistry that we actually sell through Sigma Aldrich. And broadly speaking, TDA has been into synthetic chemistry, materials processing and testing, lots of defense and aerospace, but very recently medical devices. And so the base technology that kind of let us put this nanoparticle imaging agent together is something that we pulled out of the TDA vaults for a base. And then I would like to go over the project team and my team at TDA. There's myself. I had a whole slide on me. We don't need to know anything else. My manager or supervisor, Janine Elliott, who's a PhD chemical engineer and has been at TDA for 19 years and has an expertise in polymer chemistry. Ria Martin, who is our organic chemist and is fantastic because I will write something down on the whiteboard. And we all know it's much harder to synthesize what you write on a whiteboard than it is to write it on the whiteboard. And Ria will find some way to do that, or tactfully tell me that that's not realistic and maybe here's another approach. And then Ron Cook, who's been at TDA for 29 years, almost since it was founded, who just recently retired, which means he gets to work on projects as much as he wants or doesn't want to during the week. And then this was all possible through a collaboration with Benoit Giscart at West Virginia University, and pairing up with Gareth and Sandy at DU, and then Mirigliani and Boris at O2M. Okay, so the main topic of this presentation, and probably I would say one of the main topics of this whole webinar series, is how to do this. I've grabbed a plot of the survival fraction from the paper that Howard and Boris put together, oxygen-guided radiotherapy, where they showed that what the community has been trying to do for so long was possible. You can map the oxygen concentrations, identify a hypoxic region, and treat that hypoxic region with a higher radiation dose and see an improvement in the, here we're looking at an improvement in the survival fraction with the blue line where the hypoxic boost was given, the higher radiation dose. Now, how do you take that into the clinic? And here I've used the traditional organic chemistry steps, which is usually a tongue-in-cheek way of indicating great difficulty. How do we make this transition? So I wanted to go over some of the questions and just some of the, I'm not going to call it resistance, I'm going to call it feedback that I've run into. And I think these are questions that are good for the community to think about, and there are answers to them. So the first, why do we even need PO2 maps? Why do we need to know the oxygen concentration? There are two groups then of people. One asks, why can't you just inject a naked radical into people? What's the problem? And another group who says that free radicals are bad. You can't put them into people. They're evil molecules. A question about intratumoral injection, which is the path that we chose for our imaging agent in the tumors grown in the mice at O2M, and I'll show pictures of those later. turns out that's actually a feasible route for humans. And there is an FDA cleared injectable oncolytic virus delivered intratumorally. And then my favorite, it's totally a chicken or the egg type comment. There's no clinical EPR imagers in existence. Why bother developing clinical EPR probes? And so it's actually, there are some options that you could pull from for clinical EPR imagers. So again, we have a hypoxic boost. Why do we need this? We need to generate the PO2 images. We need to target hypoxic regions. Rather than spend time trying to recover what these two gentlemen here, Dr. Halpern and Dr. Schwartz, have covered, and much better than myself. I'm directing the audience back to these talks. I know Howard's is recorded. I think Hal Schwartz's is as well. You know, go back and look at these and use them as resources. But this question I got from a scientist with the nanotech characterization lab of, why can't you just use a higher radiation dose for the whole tumor? And Howard very nicely lays out in his presentation that if you were to do that, you would risk necrosis of healthy bone or soft tissue. And the whole point of any therapeutic approach is to maximize the benefit and minimize the collateral damage. And that's really what you can do with the quantitative oxygen maps. And then Gareth and I were chatting about this, and there's another technique called flash radiology, where instead of delivering a beam of radiation over seconds, you deliver an incredibly high radiation dose in a short period of time, sub-second pulse of radiation. The jury is out on if this works better. This quote is from a recent review that there needs to be better control on preclinical studies using the flash technique in order to say that you can get away with doing that shorter time periods and higher radiation dose. But the point of using oxygen sensing probes in EPRI kind of is outside of both of these, regardless of how you're delivering the radiation. You want to be able to see where you should be delivering it to. And then the other thing that, you know, we haven't talked about a great deal is that these oxygen images take on the order of 10 minutes. And the Chicago group has shown you can watch on that time scale oxygen cycling in experiments. And so if you are beginning to get to the point where you could watch the oxygen response after radiation dose of a tumor, that's really powerful. And that's something that really resonated with the radiologists on, you know, kind of on the review panel for this technique. And so we need to paint the target. We need to make our best informed decision on the treatment, and EPR imaging helps us do that. Benoit touched on in his talk why we can't just inject naked trittle into people. The naked trittle being the deuterated Finland trittle. And it's because it is very hydrophobic and it interacts with other hydrophobic patches. If you have something that's going to disrupt hydrophobic forces in the body, that's not looking good for toxicity studies. And kind of the poster child case is this work out of Ohio State, where free trittle radical, this symmetric trittle radical, in the presence of bovine serum albumin, BSA, that damped the trittle signal. It squished it out. Now, what's interesting is they could recover the trittle line width. So this interaction was not destroying the trittle, which makes me think it was just slowing it down quite a bit, and that some relaxation measurements are in order. But what do we do for the mice? Well, we make molecules like OX63, or we deuterate it, and it becomes OX71. And it makes it much more hydrophilic, but we pay a price both in terms of line width and also frequency response when we do this. The symmetric trittle, hydrophobic one, has much less response of its relaxation times, much less penalty that you pay from 9.5 gigahertz down to the imaging frequencies sub 1 gigahertz. So you can either change the radical chemistry, or you can try to separate it from the body environment to cut off that interaction. And I just, as an aside, want to point out, I can remember coming up through the Eaton's group, and the gargantuan task of getting a trittle molecule, let alone many trittle molecules. And Benoit has his website here, this immrprobes.com, where he lists everything out and, you know, says, hey, contact me and, you know, we can work together, or I can make you some. And that is phenomenal, and phenomenally useful, not just for the people who are in the field, but for bringing new people in and saying, well, you know, here's a way you can get some of these different imaging agents. You can't put free radicals into people. Up here in the upper right, oftentimes what I'll like to do is I'll do a quick Google image search to see how, I don't know, the society, the group think feels about something. This is one of the first pictures that came up. We have the healthy cell. It's scared because there's an evil free radical right next to it, but there's a really happy smiling antioxidant to the left of that. A free radical, we think of things like hydroxide radical or superoxide radical. It's a small molecule, it gives up an electron easily. An antioxidant has a chemical structure where it can accept an unpaired electron from a free radical, but it can hold it within that chemical structure safely. And in fact, EPR probes, we don't want them to give up their free electron. That's what's reporting on everything. And so they are designed to hold on to it. And you should not be surprised that a chemist is telling you the house you put the electron in matters for what it is. So in fact, people hear free radicals when we talk about EPR imaging and they think this, but yeah, aside from hydrophobicity issues we just mentioned, they're a lot more closely aligned with an antioxidant. And in fact, this is our old friend Tempol, one of the oldest nitroxide radicals, is currently heading into phase 2 clinical trials for oral administration as a treatment for cerebral cavernous malformation. So this is being driven by a company which I have no stake in Recursion. They're in Salt Lake City, I believe, in Utah. But I have been watching them because this Rec 994, which is Tempol, was one of their first drug molecules identified and given orphan drug status for use in this CCM. And essentially in cerebral cavernous malformation, you have blood vasculature abnormalities. The blood vessels are thin and leaky. And you have reactive oxygen species that are being generated but not dismutated. And so Tempol is used as an antioxidant to neutralize the reactive oxygen species. Now, oral administration is not intravenous injection or intratumoral injection, but it is one step closer to a, you know, I mean, this is an imaging probe. It's not as good as the Trittle, but it is something you can image with in a body approved by the FDA, getting away from this radicals are bad business. And you can put protected free radicals into people. Everybody circle your calendar for September 10th when Perianan Kupasamy is going to give a webinar. I have this single slide because Koop's work has been exciting to watch for many years now. Koop's has gone the route of encapsulating kind of macro-sized or millimeter-sized. This was a triaromethyl radical, a Trittle. This is the lithium phthalocyanine or the butoxy radical-based radical in PDMS, and has actually inserted them into tumors surgically and read out the oxygen in a spectroscopy, not an imaging sense, and has just recently published, and I'm sure he'll talk about their observations with the first clinical trials. And this is very exciting. You know, MRI has 7 different commercial gadolinium contrast agents alone that are available to do imaging with, and a number of different imaging sequences. I hope for, and I wish for, a clinical EPR imaging that both would cross that bridge, but then we also develop a number of different probe sets that could gain FDA clearance to help bring the really powerful information that EPR imaging can provide to people. I certainly want to say that if as a much older man, if I have grandchildren and one of them was to have a cancer in a solid tumor, I would want to know that I had tried everything I could to bring this technique, which could help them in the future, across that divide. Something I learned, I thought might be a difficulty, was this use of intratumoral injection, but the FDA cleared solution is Imlygic. It's an oncolytic virus. It both targets and causes destruction of the cancer cells by dividing, as viruses do, and bursting, but it also summons the immune system to the tumor to help destroy it. And if you read the directions for administering the Imlygic, single insertion point, multiple tracks, actually what they're advising is you go in one spot and then kind of spread out from there. Our intratumoral injection that we did on the mice in the imaging portion of this was either different tracks from all around or the same tracks all parallel. But this is already cleared for human use and would be an appropriate vehicle to deliver, say, an EPR imaging agent for tumor oximetry. And this is my favorite one. Why do you develop a clinical EPR imaging agent when the commercial hardware doesn't exist? It's definitely, even the program director for my project said, I recognize this is a chicken and the egg problem, okay? You need one and the other. But in fact, you know, I think Hal mentioned this when he gave his presentation, there's quite a bit of knowledge that Hal has amassed and the community has amassed on how you would do large animal and then eventually human imaging. And ClinEPR does advertise in vivo clinical EPR systems that look something like scaled up versions of our small animal. So that was a really easy answer. You could say, actually, technically they are available. And that then says, well, we must have imaging agents for them. You could do an Apollo 13 approach. This is a fun game. We used to play at NIST quite a lot because whatever you are at NIST, whatever you came as, you also become amateur electrical engineer. So you were a physicist, you were a chemist, it doesn't matter, you're that, and also amateur electrical engineer. And you would get points for how few parts you could use to do this. We need to make this fit into this hole using only this. I would actually not get many points for this suggestion. But you know, if you're looking at a commercial, this is a Siemens commercial 7T MRI, which has all the electronics for 298 megahertz, which is one of the EPR imaging frequencies. You're going to have a little bit of resistance trying to drop a supercon magnet to 10 millitesla or 15 millitesla. But as was pointed out by my former colleague at NIST, Carl Stupik, they also have Siemens has in their product line this magnetom-free max, which is designed to operate down to 0.55 Tesla. And the gradients are designed to run without cooling. And in fact, it's kind of counterintuitive, but when you do imaging for EPR imaging, you go really slow or static in your gradients. The electron relaxation times are too fast to do the singing gradients tricks they do in MRI, where they're just flying around. We don't have that much time. It becomes easy then to just set the gradient, collect the image, set another gradient, right? And that's how it's done. It's actually a lower, kind of a lower ask of the gradients than MRI. So, could I do this at TDA? No. But Siemens and GE, they all have sites. They have research sites all over the world where people could do this, and they would have access to these product lines. And I would not get any points for this in the NIST game because I did not have to use any pencil, erasers, rubber bands, or paper clips to make it work. And then the kind of longest term route is that there is a funding mechanism in the government, the technology transfer grant. And that is specifically for pulling information and knowledge that's developed in the university setting out in the commercial sector. And so this is a picture of the DU 250 megahertz magnet that I spent many hours on during graduate school. There's a sister system, which is larger, and I would call a small human imager in Chicago. But this kind of design exists. You could look at pulling out like that. And you could even look at things like this bottom right picture shows a hyperfine portable MRI, where instead of having the main field magnets vertical, they've flipped them and they're horizontal, and that's better for access. You can get head access and arm access and leg access. This is a low field MRI, 64 millitesla. Again, I have no stake in hyperfine other than I think the technology is cool. And this kind of form factor, which has kind of received some acceptance already, is something that we begin to think about what might a clinical system look like. All right, so the radiotherapy guided by the oxygen images is valuable, and it would improve patient care. Commercial EPR imaging does exist, and there are more options in the line. And the form of the radical matters. And if you can adjust the form of the radical either through chemistry or by putting it in a package which is amenable to use in the body, you know, we can get around that hurdle. And this part, separating the radical from the body environment, is really what we're after in the work. Some of the work I'm going to talk about from our recent NCI project. So this was a collaboration, TDA, West Virginia, University of Denver, O2M, and then also we had a collaborator for some of the imaging, electron microscopy imaging at the School of Mines, which is just 10 minutes up the road from us here. So we want to put our paramagnetic imaging agent on a nanoparticle. We want to do polymer encapsulation. We need the O2 to be able to pass through that polymer layer. And we need similar PO2 statistics for a tumor as you would get from the traditional Trityi. And the other caveat here is this was a proof of concept study with demonstration in 9 months time frame. And so again, I have to thank my collaborators. All of the pieces worked really well together to kind of generate some proof of concept data. So we had nanoparticles. These are scanning electron micrographs from mines. This is one picture where we see in blue here, there's a big guy, he's around 600 nanometers. There's lots of these smaller guys, you know, around 90 nanometers, and then a good chunk of sub-90s, and some debris here from the sample, and it just was dried out on a gold backing for the SEM. And then this is just a zoom in of these guys. This is what we were working with for this project. We also were able to do particle tracking analysis to confirm kind of the size distribution over a larger sample. And so particle tracking analysis, you have your samples of nanoparticles in a little cuvette, and you shoot lasers of different wavelengths at it. And depending on how the wavelengths of those lights interact with the particles, that interaction is dependent on the size. And so I'll call your attention here to this one little particle here that's kind of getting a blue-red reflection is larger than these ones here that are interacting and scattering primarily with the blue light. But this gives us, you know, a good measurement. We're sitting around 90 nanometers for, you know, half of our nanoparticles are smaller than that. And we've got a relatively narrow distribution of them. We worked with Benoit and were able to acquire some Trityi and just run the very quick in air versus in nitrogen and get a very nice line width change. What we're actually interested for the imaging is the relaxation times, the electron relaxation times, and specifically the T1E, as Howard's group at Chicago, you know, showed previously that it is less sensitive to concentration effects than the T2E, the electron spin, but relaxation time. If you're less sensitive to concentration and you're a little more sensitive, or there's less going on to interpret, you can attribute more of the change to oxygen. And so, okay, it works fine when it's in solution. This is just free, and PBS, pH 7.4. When you attach it to the nanoparticle and when you attach and encapsulate, you see the T1E increases by 5 to 6 microseconds. And so we step back and we say, does that make sense? I was trained up in relaxation times and relaxation theory. Usually I bring this up and it causes people's eyes to roll back. And it dawned on me that what I don't ever say, what I'm saying now, is that when I want to look at a molecule and I want to consult the literature about what drives the T1E and the T2E. I don't do this literally, but figuratively, I put on my electron relaxation vision specs. They're not quite as cool as these X-ray specs that this gentleman's wearing here, but that's the idea. I'm looking at the molecule and I'm thinking about what changes in the environment and the molecule are reflected in the electron relaxation times. And so I've drawn a little cartoon here of the unpaired electron interacting with deuterium on the symtrittle, a intermolecular dipolar interaction. There's also interactions with nuclei in the solvent sphere around the trittle. And there's a couple of implications from these interactions. The first of which is that these inter and intramolecular interactions are modulated by how fast or slow the radical tumbles in solution relative to 1 over the frequency of operation in radians, or 1 over omega. So symmetric trittle by itself in solution is tumbling about 290 picoseconds, reorienting in solution. At 9.5 gigahertz, the 1 over omega is 17 picoseconds. The difference between this value and this value up here are very different. And as you go to lower and lower frequencies, the 1 over omega value approaches this tumbling correlation time of the trittle, and you start to lose. So this is a 1 over T1. Going up on this plot is bad. It means your T1 is getting shorter. And that effect is much larger. It's not shown on this plot, but if you were to go to look in this paper, it's much larger when you have the OX31 or the OX71 or the OX63 radicals, where you have more protons out here and no deuteration. But what it does mean is that at some very long tumbling correlation time, you move well past these low frequency 1 over omegas, you might expect the T1E to become independent of frequency. And it also means that you need to know, if you say, I'm making my T1E longer because I stuck the trittle on something, you need to know by how much. There's a region here of, we'll look at the protonated trittle, a region here where increasing the tumbling correlation time will actually shorten your T1. This is increasing the rate, so it's shortening your T1. And then if you keep moving past that, it will get long again. For the symmetric deuterated trittle, we actually have to look at the tumbling correlation time of the solvent, because there's so little interaction due to the deuteration. And so how do we take that back and look at our relaxation times and think about what the trittle's doing on the nanoparticle? We were able to run some experiments where we could also attach a proxyl radical, which is very established for working out changes in tumbling correlation time versus changes in the CW spectra. And that little guy, smaller than the trittle, was at a tumbling correlation time of 3.6 nanoseconds when it was bound. And so the trittle is at least that slow, maybe a little slower. If you were watching Benoit's talk, you would say, Josh, this is an excellent application to use Benoit's new C13 labeled trittle. And I'm trying to think of an experiment to set up now to do just that. But we're at a long enough tumbling correlation time that this makes sense, that it's on the nanoparticle. We haven't gone far enough to lose our frequency dependence. So this is measurements at, actually, I think it's closer to, Mirignani and I were just talking about this, 720 megahertz instead of 750. But the T1E in the absence of oxygen is 7.5 and 4. But when you look at the measurements at X band, and we've also tracked this by other analytical techniques, this is showing that we have the trittle radical on the nanoparticle. So how did we assess that encapsulation went through? So here is, this is your plan, and this is reality down here on the bottom. The plan was to just do straight transmission electron microscopy. And the reality was we had COVID shutdowns, and then we had an electronic board fail on the Talos TEM at Mines, and then we had the software quit and erase the user database. We're on track now to get some TEM images. But in the meantime, we did a couple of tests as just stand-in. One of those was to look at the change in the size of the nanoparticle before and after encapsulation. And this change of about 90 to 114 nanometers roughly corresponds with the theoretical thickness that we calculated we should be getting. We also did a test where we know that free symmetric trittle in solutions of bovine serum albumin will squash the signal of the trittle. This is free trittle, and this is just in air. This was just a quick test I ran recently at X-band. And then the blue trace is 50 micromolar BSA. And in fact, if I had not blown this up by factor of 10, you would have just seen a mostly straight line across, blue line across here. It really depressed, broadened the line width. If we make the same test with 2 different batches of our encapsulated imaging agent, we get some increase, but not nearly as much as the free trittle. So this is 20% and 50% versus 140% increase. We have some optimization to do for batch to batch reproducibility. And we are awaiting the final TEM images to measure it exactly. But we have 2 pieces of information that say that that is likely true. Okay, we're gonna run through really quick a couple of images. This was the sequence for the imaging at O2M. In here, this is not injection of the imaging agent, it is inoculation with tumor cells kindly provided by Howard's lab. And then growth of the tumor on the mouse leg, the mouse here in the imaging bed, MRI to register the tumor position, OXO71 delivered for PO2 imaging via intravenous route, a wait time for 60 to 90 minutes for it to clear, and then imaging on the same tumor on the same mouse with the encapsulated imaging agent, and then we compare the statistics. There were 2 different tracks tried for the imaging. We didn't seem to notice a big difference on how the imaging agent was delivered, but this is something we might revisit. The signal's noise of the RADI-sense image was low, but still usable. We were able to image 5 mice in total. The difference between something like the PO2 images for mouse number 1 and mouse number 4, which I'll show in the next slide, were we did a volumetric concentrating of the encapsulated imaging agent, which gave us a little bit better signal to noise in mouse number 4. So if we look at the EPR signal amplitudes for mouse number 1, they're not very good, they're not very high. If we look at the oxygen images for mouse number 1, so in the middle here, this is IV delivered, and in the bottom we have intratumoral injection of our O2, our encapsulated imaging agent. It gets a little bit better. There's much better agreement in the amplitude, and there's a higher amplitude in the last slice here of this mouse. There is some thresholding that O2M had to do because of the lower signal to noise of the imaging agent. Thankfully, the imaging agent is not optimized in terms of labeling, so we have a lot more room we can go towards this better signal to noise. But if we look at the statistics and the PO2 statistics that we can calculate from the regular trittle delivered intravenously versus the encapsulated agent, we see that as we increase the concentration just by decreasing the volume, we're getting better agreement in a number of voxels that are actually used to calculate the PO2. The median values of PO2 are a bit lower when done with the IT injection of the encapsulated agent. Although, even if you do IT injection with OXO71, the regular trittle, those values will be a little bit lower than IV injection. So a kind of a next test would be a head-to-head intratumoral injection comparison. But this regular trittle delivered by IV was the standard way. And that was really the goal of this proof of concept study, is that you did not have to make special allowances for the encapsulated imaging agent. And then we have some T1 and T2 slope values here that are comparable for the anchored encapsulated agent versus the free trittle in solution. So it's certainly true that we need to improve the signal to noise of the encapsulated imaging agent, but we were able to retain oxygen sensitivity and calculate quantitative PO2 maps with it. So 2 summary slides here. The preclinical evidence, there are decades of excellent preclinical evidence about why EPR imaging is beneficial and necessary. The hardware is there or close. Radicals could be used in people as part of just spectroscopy, treatment of disease with the temporal and the cavernous malformation or imaging. And again, we don't get the problems that look this clear. Here is the screw, here is the screwdriver, insert and turn. And so, you know, it's there are many tasks to be overcome to move EPR imaging from preclinical to clinical work, but it's a worthwhile cause.